So thank you, everybody, for giving your time this morning to our awardees. I'm excited to present at least two of the three that we're going to have over the course of the think tank. I particularly want to just ask everyone to give a round of applause to all our hardworking Beacon staff and everybody who took the time and effort to review these grants. It's a lot of effort. It's done entirely voluntarily, and it really elevates the entire science that we're presenting to you today. So I've asked everybody who reviewed, please stand up and give everybody. Okay, so we'll get going. The first presenter is Dr. Raie Bekele. Hopefully I pronounced that right. He is a PhD in structure of radiation oncology at the Dana-Farber. His short bio states that he's a bladder cancer biologist with a research interest in DNA repair processes and MAP kinase signaling pathways. He's originally from Addis Ababa in Ethiopia. Several childhood experiences inspired him to do science and moved abroad to receive his bachelor's in biochemistry and cell biology at Jacobs University in Germany, followed by a PhD in biochemistry from the University of Alberta in Canada. He then moved to the U.S. for his postdoctoral training, first at the Mass General and then at the Dana-Farber. And he's currently an instructor in radiation oncology at the Dana-Farber and Harvard Medical School, and his project is entitled Dissecting the Interplay Between MAP Kinase and PPR Gamma Signaling in Bladder Cancer.
Thank you very much for the warm introduction. Can you all hear me? Yes. Good morning, everybody. I know it's an early start, so I appreciate you all for being here. Let's see if this thing works. All right. The title of my talk is PPR Gamma, the Rogue Conductor, Broken Harmony, How MAP Kinase Alteration Drive Bladder Cancer Progression. This is a symphonic title. I went for that, but don't worry, there will be no singing today for this presentation. In the orchestra of cellular activities, sometimes conductors like PPR gamma could go rogue. And when this happens, there could be disarray. Today, our focus will be on PPR gamma, a pivotal player in the cellular symphony. And when it gets into disarray, the cells become chaotic and become cancerous. We will also be exploring MAP kinase alteration and how they contribute to this pathway. PPR gamma is the conductor. PPR gamma is a peroxisome proliferating activated receptor gamma. It's a master regulator of lipid and glucose homeostasis. In addition to its role in metabolism, PPR gamma has a unique role in the bladder, and it activates luminal differentiation program. What you see here is the basal stem cells differentiate into intermediate cells, and they become specialized cells, forming the urothelial barrier through the activation of the PPR gamma signaling pathway. In addition to this luminal differentiation program, PPR gamma is also responsible in modulating inflammatory responses. So how does PPR gamma become rogue? Before we get into that, this is a very nice demonstration showing that PPR gamma cross-tissue expression. And what we see here is that bladder tissue, along with breast and adipose, are one of the highest expressors of PPR gamma. This shows the importance of PPR gamma in bladder. And another is the single cell data of prostate tissue. And as you know, the prostate has some urothelial cells, and the urothelial clusters you see there also have high PPR gamma expression. This shows the predominance of PPR gamma in bladder tissue, highlighting its importance there. So one way this protein, this vital protein that orchestrates PPR gamma differentiation, bladder cancer differentiation, becomes rogue is through aberrant activation. To delve into this, we can look at a population-based study concerning pioglitazone. Pioglitazone is a PPR gamma potent activator. Sorry about that. And it's also an anti-diabetic drug that's given to patients orally. And what we see is that patients that have gotten pioglitazone as an anti-diabetic drug develop bladder cancer. This clearly shows the link between aberrant PPR gamma activation and bladder cancer development. Another way PPR gamma could be aberrantly activated is through copy number amplification. This is the data, copy number data from the TCGA. As you can see, a subset of bladder cancer patients, muscle-invasive bladder cancer patients, have copy number amplification of PPR gamma. So one way PPR gamma could be aberrantly activated leading to bladder cancer could be through copy number amplification. Now, shifting a gear a little bit to MAP kinase signaling pathway, what we see is that in chromosome 3P, we have PPR gamma adjacent to RAF1 gene, which is a MAP kinase signaling protein. And aberrant chromosome activation leads to copy number amplification of both genes. As you can see, we saw that patients who have copy number amplification in RAF1 also have copy number amplification of RAF1 as well. So you can see they are both co-altered. What we also see is that MAP kinase alterations such as HRAS, NRAS are also mutated. And this makes up 24% of the total bladder cancer population, showing the importance of MAP kinase alteration in bladder cancer. So as we discussed earlier, PPR gamma through aberrant activation or copy number amplification could lead to bladder cancer, and it's also co-altered with the MAP kinase pathway. So what's the MAP kinase signaling pathway? If fibroblast growth factor, epidermal growth factor bind to their cognate receptors, receptor tyrosine kinases, and activates RAS. There are three types of RAS: HRAS, KRAS, and NRAS. They bind to the RAS binding domain and change the conformation of RAS, leading to the activation of MEK and ERK signaling pathway. What that does is it activates growth and proliferation differentiation signals. Another interesting thing I want to mention here is that there is an intimate connection between RAF1 and HRAS, which you also have seen are one of the most highly altered genes in bladder cancer of the MAP kinase. The reason that being is that BRAF has a predominant binding to KRAS, whereas H and NRAS prefer RAF1. That's why we see in bladder cancer this RAF1 and HRAS connection, where these two proteins are altered. So it's very important. So exploring MAP kinase alteration in bladder cancer TCGA, what we see is across different TCGA patient cohorts, we see that RAF1 amplification is highest in bladder. So if you want to study RAF1 amplification, bladder cancer is a good model system to study that. And you see here, and there is a fish looking at copy number alteration of RAF1. And what you see is that this is a patient sample from Dana-Farber Brigham institutional cohort. And what we see is that a huge amount of copy number. You can see here, the pink dots indicate all the copy numbers there. And when we compare the alteration frequency between our cohort and the TCGA, we see similar alteration. So to investigate this, what we did was we looked at RAF1 expression across different bladder cancer cells. And what we find is two cell types that you can see here in the RNA expression, UMUC9 and 5637, have a very high copy number amplification shown through the RNA expression. And you can see from the Western blot clearly, these two cells have high RAF1 levels. What we did next is we looked at CRISPR screens, looking at a knockout of the RAF1 gene across different bladder cancer cell lines. And what stand out, the ones that are dependent on RAF1 are the MAP kinase altered cell lines, RAF1 altered cell lines, HRAS, and NRAS. So you can see that when there's MAP kinase alteration, taking out the RAF1 protein is deleterious to the cells. We also did genomic targeting of RAF1, and what we see is that in the cells that have RAF1 amplification, genomically targeting RAF1 leads to their cell death. We also see that MAP kinase activation of RAF and MEK is abrogated when we inhibit or when we take out RAF1. Next, we did a combination study whereby we combined RAF and MEK inhibitors, and we see that MAP kinase altered cells are killed or are very sensitive to this combination inhibitors, whereby the RAF wild-type cells like J82 here is spared, which you can see the importance of RAF inhibition in killing MAP kinase altered bladder cancer cells. We also did took RAF1 amplified patient-derived xenografts and preclinically tested them with a combination therapy. And what we see is a significant reduction in tumor volume and tumor weight, indicating that this could be a good novel target for bladder cancer. So we've discussed that PPR gamma is altered and it's linked to bladder cancer. And we see that MAP kinase alterations are drivers in bladder cancer. So how are these two proteins linked? To explore this interplay of these two crucial signaling proteins in bladder cancer, we used a bladder cancer RT1112, which has a nanoluciferase, CRISPR knocked into the endogenous locus of a gene called FAB4, which is PPR gamma regulated. And what you see here on the green is the control cells having high amount of PPR gamma activity. And then when we inhibit with a PPR gamma inhibitor, to your right, you see the PPR gamma inhibitor completely blocks its activity. And what we see is that RAF and MEK inhibitors are able to decrease PPR gamma activity. This indicates that inhibiting MAP kinase pathway not only blocks the RAF MEK ERK signaling, but also blocks PPR gamma activity. And what you see on the right side of the panel is data from our collaborators at the Broad. And here again, you see rosglitazone, which is another TZD drug, which has a very high PPR gamma activity. You can see activating the cells will lead to high amount of PPR gamma. But when you see the other inhibitor, which is in clinical development, PPR gamma inhibitor at the bottom, SR1 compound, and you can see the trametinib in the yellow, showing that there is equal amount of depletion. So both the PPR gamma inhibitors and trametinib are able to decrease significantly the PPR gamma activity. So in this way, PPR gamma and MAP kinase are linked. So what we did next was to do the opposite, whereby we took those cells, the RT1112 cells that have nanoluciferase, and we overexpressed RAF1 gene. And what we see is that when we overexpress the RAF1 gene, we were able to have a significant increase in PPR gamma activity. And this is also going when we had increased amount of pioglitazone, the other TZD I mentioned, that cause bladder cancer. And we can see the RAF1 amplified cells are having a higher level of PPR gamma activity. So we wanted to find out, okay, when we inhibit PPR gamma, we decrease activity, and when we overexpress RAF1, we also have an increased amount of PPR gamma activity. So we wanted to see how this happens. So we took cell lines like HEC293 T cells, which are not bladder cancer cell lines, but they don't have a lot of PPR gamma. So when we overexpress RAF1 in these cells, what we see is an increase in PPR gamma levels. So we think RAF1 regulates PPR gamma activity through increasing its expression. And we also took cells, BBN, which we got from our collaborators, the Kim lab, and when we overexpressed RAF1 in this carcinogen-derived basal mouse bladder cancer cells, what we can see is an increase in PPR gamma levels. So this together shows that MAP kinase signaling through RAF1 increases PPR gamma activity through increasing expression. So then we moved back to the cell lines that have RAF1 amplification. In this case, 5637 and UMUC9. As I've mentioned earlier, these cell lines have copy number amplification, like a lot of copies of this gene. And when we inhibit RAF1 through LXH, the RAF inhibitor, or MEK inhibitor, what we see is a depletion of this protein. This is very significant because this is our copy number amplified cells. So we were able to completely deplete, not completely, but significantly deplete PPR gamma expression. As I mentioned, one of the roles of PPR gamma in the urothelium is to modulate inflammatory responses. So what we did in the right panel is to look at inflammatory responses. And when we inhibit with PPR gamma, we see change in cytokine expression. And we were able to phenocopy this by inhibiting with RAF1 and MEK inhibitors. This then shows that the intimate links that we have between these two signaling proteins. So this is one of our newest data. And what we did here is, again, we took cells, these are luminal cells, UPPL 1541, which are PPR gamma activated. What you see here is we do not expect immunotherapy, which is approved in bladder cancer, to work in this luminal bladder cancer, mouse bladder cancer cell line. And what we see is that when we inhibit with a combination of RAF and MEK inhibitor, we were able to slow down the tumor because potentially it's a PPR gamma activated tumor. We also used a JAME model of HRAS, which has HRAS Q61 mutation. We got this mouse model in collaboration with the PAN lab. And what you see here is we have a wild type mouse, which is there, which has a small bladder, mouse bladder, and then there's an SV40 uroteller expression of SV40 mouse. And when you cross it with a mouse which has HRAS mutation, then you get this bladder tumor. But the SV40 expressing uroteller expressing mouse tumor only has cyst, carcinoma in situ, and the HRAS mouse only has bladder hyperplasia. But when you cross them, you get a bladder tumor within 3 to 5 weeks. And in this tumor model, when we inhibit RAF1, when we inhibit the RAF and the MEK signaling pathway, you can see that there's a huge depletion in the PPR gamma levels. There, you can see HRAS model clearly showing a high amount of PPR gamma, as you see from the pink staining there, which is for PPR gamma. But when we inhibit it, you can see a decrease of that. This shows that MAP kinase is active in this models, and targeting them with MAP kinase inhibition could be a novel treatment strategy. Finally, just to summarize, the MAP kinase symphony, where the rogue conductor PPR gamma and its upstream regulator, as I've shown you through a series of data, both in concert drive bladder cancer progression. The use of MAP kinase inhibitors can just restore this harmony, paving the way for new targeted treatment strategies. I'd like to emphasize that so far there are no MAP kinase inhibitors in clinics for bladder cancer patients. So we hope that advancing MAP kinase inhibitors would be a novel treatment strategy. I'd like to thank everyone. Special thanks to my mentor, Dr. K. Mao, sitting over there. And thanks to BCAN for funding my project. Thanks to the various collaborators for making this project a success. And a lot of my colleagues and members are listed there. Thank you very much for listening to me. If you have any questions, I'll be happy to answer them.
Great presentation.
Thank you.
So I have 2 questions. So the first question is, I've never been able to explain the co-expression of PPAR gamma and FGFR3 that we see in a lot of these luminal tumors, including upper tract. And as you know, FGFR3 interacts intimately with MAP kinase. So have you been able to look at that in your system? I mentioned that because, for example, one of the cell lines that uses RT112 has an activating FGFR3 alteration. So one could imagine that is something to look into in those cell lines. But also, we see this a lot in human cancer, so I think it's a really important point. The second question is whether you've observed any luminalization or luminal phenotype in your basal, in the basal cell lines, the BBN-induced cell lines, because as we all know, PPAR gamma is a master regulator of the luminal phenotype.
Very much. Those are great questions. I will answer the second one. We're actively looking at the luminalization of the basal proteins. One of the luminalization patterns is you see the increase in PPR gamma levels. So that could potentially indicate that they could be they're becoming luminal. For your first question, yes, I agree. The RT112 have FGFR copy number amplification. And FGFR, one-way bladder cancers, drivers like FGFR activate the MAP kinase signaling pathway. So if there's FGFR mutation, we don't see maybe RAF1 amplification. But those two go in together because when we inhibit RAF inhibitors are also successful in the FGFR altered setting as well. The reason that being is that they signal through that pathway.
Thank you.
Matt Sheaver, Thomas Jefferson University. Very nice talk. Thank you for sharing. Do you have any ideas as the mechanism by which when MAP kinase pathway is altered, you get a change in PPAR gamma levels?
Could you repeat that?
So when you manipulated MAP kinase, you had a different expression level of PPAR gamma. Do you know what is making that happen mechanistically?
Yes, we're actively working on that. We think that MAP kinase signaling, specifically not RAF, but like all that signaling together, leads to the synthesis of PPR gamma. So MAP kinase is important in the transcriptional synthesis of PPR gamma protein.
Okay, and then I don't study metabolism, but I was wondering if, since you see this switch in PPAR gamma, if you are looking at whether that it changes them to a differential lipogenic or some other metabolic thing that might uncover another potential way to target the cells.
Yes, that's a very interesting question, and we have not specifically looked at that, but we do believe that there could be this metabolic vulnerability of these cancers because of the increased PPAR gamma levels.
Thank you.
Thank you.
You were first.
Yeah, I'll follow up on that question. So I think KRAS obviously induces metabolic changes that are pretty anabolic. And you think of PPAR gamma, while you have amplification, you do need the ligand to really turn on the nuclear receptor. So I'm curious, is there actually a intersection where RAS induces metabolic changes that may upregulate the ligand, and therefore, when you have more PPAR gamma around, actually super stimulate? Because as you showed with the pioglitazone or rosiglitazone data, having the ligand really amplifies the transcriptional outputs. So I'm curious if the sort of the ERK pathway and PPAR gamma may link in that way.
That's a good question. That would be what I will do my research program on when I become independent.
All right.
But that's a great question. PPAR gamma activators like rosglitazone and pioglitazone, they are like very, very potent. The natural agonists do not get to activate PPAR gamma to that level. But what we think is that in bladder specifically, there could be some lipid factors that could potentially activate PPAR gamma, and we don't know that. Generally, it's thought eicosanoids and fatty acids of different kinds can activate PPAR gamma. So we don't know, maybe huge fatty acid intake or cell death of the urothelium could lead to this release of fatty acids activating PPAR gamma. So we don't know what this natural ligand is and how the KRAS is involved in that process, but there could be a link. Thank you for that question. Any more questions?
Hi, this is Bob Schreiber, patient advocate. In one of your early slides, you cited a drug that I think was for diabetes, anti-diabetic drug. And so I was curious from a patient perspective, was that drug protective or possibly leading to bladder cancer? And if it was leading to bladder cancer, I was just wondering if we should be putting out a warning to patients taking the drug, because often we're dealing with older patients. A lot of bladder cancer patients may also have diabetes. I'm just wondering if that's something that should be done to help protect patients.
That's a very important point. The pioglitazone has a black box label warning on it here in the US, and in the Europe, it's banned for anti-diabetic drugs. So yes, I mean, it has an effect in regulating glucose homeostasis for diabetic patients, but long-term usage, specifically pioglitazone, can increase the risk of bladder cancer. So there is a controversy whereby, depending on the treatment and how long the treatment is given, there could be an increased risk. So there could be maybe other ways of modulating or decreasing the diabetic issue without having to increase bladder cancer. So actually, it could be contraindicated for bladder cancer patients.
Thank you.
Great. Thank you very much.
All right, fantastic. So our next awardee is Laura Bukavina, who is a urologic oncology fellow at Fox Chase. She writes that she's a fellow at Fox Chase Cancer Center with extensive research focused on the microbiome and its influence in bladder cancer treatment response. Her research has centered around combining her experience in cancer epidemiology and immunology to expand the mechanistic understanding of the microbiome and its relationship with the tumor microenvironment. And while her previous research has established key microbial networks that inhibit treatment response, the goal of this project is to modulate treatment response through personalized microbiome interactions. And the title of her work is Anti-Tumor Activity of Lactobacillus rhamnosus GG in a murine model of bladder cancer.
Hi, everyone. So I'm on stage, but I would like for Phil to get up as the mentor who deserves equal credit for this data. Phil, go ahead and get up. I was a fellow at Fox Chase, recently graduated fellow and starting my first job at Case Western as a surgeon scientist in urologic oncology in about 10 days. So I want to discuss the aims of the project. We're going to start with some of the introduction of our preliminary data and how we got to where we are now. Describe the data from our urine microbiome analysis across multiple institutions, and highlight our current results of LGG modulation in bladder cancer model. So briefly, we know that bladder cancer is a global disease, about 500,000 incidents, with about 212 deaths every year. This is 2020 data. We know that BCG currently is standard of care for non-muscle invasive bladder cancer. It's the only bacteria that's been FDA approved as immunotherapy since about 1970s. It's been in use. It's no longer in use for other multiple diseases. It was previously been utilized in melanoma. However, there's alternative treatments across multiple disease. However, we still use BCG despite really not knowing how it works. We know bits and pieces of it. We know that it's CD8 independent. We know it's CD4 dependent. We know that we used to think that drainage to lymph nodes was vital for its ability to introduce memory T cells. We know that that's no longer the case. We also know that it's driven by interferon gamma. However, a lot of this information is bits and pieces. We still really don't know. Because we don't know, we also don't really understand why people are refractory and why people don't respond. By having limited information about BCG, we really can't pinpoint who's going to be a responder, who's going to be a non-responder in disease. We also are lacking that CD8, that critical CD8 response in BCG. By lacking that CD8, we have limited cytotoxic capability and are focused on BCG treatment despite not really knowing the mechanism well. So we started our data based on preliminary urine data from Case Western of 150 patients and looking at responders versus non-responders. What we found is that lactobacillus was significantly was present in a lot of the responders in addition to healthy patients. And one might say lactobacillus, it's a contaminant, females have it in their vaginal tract, but the funny thing is these are 90% males. So what is it doing there? And then we have a hate-love sort of relationship with these microbiome characterization studies because it's a pretty picture, but what does it actually say? So then we went on to a different cohort. Perhaps there's contamination, perhaps the collection, perhaps the PCR was contaminated. So we collected data from a different cohort, geographically different, completely separate cohort. And we still found that lactobacillus was the only predictive factor, predictive bacteria for response in these patients. So then we reached out to our friends, Jillian, who's in the audience, as well as Alexander Wyatt's data on tissue, on muscle-invasive bladder cancer tissue and metastatic ctDNA tissue. And we wondered, okay, it's present in the urine, it's being shed. Is it actually present in the tissue, in the actual bladder tissue? And it is. Actually, these are noted by the dark blue, and every sample of muscle-invasive bladder cancer was present across the board. What's even more impressive is that when those patients metastasized, lactobacillus was present at every metastatic site. So not only was it present in urine, present in tissue, but it was also present in metastatic sites. And these are metastatic sites that we consider to be fairly sterile. Granted, there's contamination from the biopsy sites. However, none of these contaminations are consistent with lactobacillus. So again, strong evidence that it's there, it's present. What is it doing? So we looked into research what else has been done in lactobacillus, and even as early as 1970s, people have known that lactobacillus induces a strong CD8 response, particularly in a lot of colon cancer studies in microbiome. It's been shown to be a great probiotic. It's the only probiotic that came close to FDA approval as preventative for cancer prevention, and it's currently undergoing FDA review. So we know there's evidence to show that it's CD8 responsive. So what does it do in bladder cancer? So our first aim was to evaluate bladder cancer tumor regression after LGG intravesical installation in mice, and then compare tumor growth and stage of LGG versus BCG versus gemdosi. We did include gemdosi as one of the arms because of the new evidence, as well versus our control. So the way we looked at tumor regression is both pathologically but also ultrasound imaging. So after about 12 weeks of BCAN, which is a carcinogen that the mice drink, at about 16 weeks they develop tumors. We're able to measure these tumors using ultrasound, and every week after their installation for 6 weeks we measure tumor volume. So you can see that the blue is the LGG, the yellow is the gemdosi, and then the other two are BCG in the control arm. You can see that gemdosi and LGG has significant tumor regression, very similar to gemdosi. We always knew that BCG was not an ablative drug, so we never expected it to really cure mice. These mice are not undergoing TURBTs. We don't have endoscopic equipment for mice to debulk them. So we didn't expect the BCG to essentially shrink these tumors. However, it was impressive to see that chemo, which we expected to potentially shrink the tumors, and LGG was intravesical therapy, were able to both shrink tumors pretty significantly and very similarly. So then we looked at staging, pathological staging of the disease, and 4 out of 10 in both gemdosi and LGG arms had a complete response on path. And this is compared to BCG, only had 2, and none in our control arm. In addition, if you look at staging of disease, LGG and gemdosi had more T1 disease and less T2 and above disease, noting that some tumors regressed in volume, and perhaps were T1, and some tumors were complete response based on this data. But I can tell you that the tumors that were listed in LGG and gemdosi, who were the T3 in LGG, was actually squamous and did not respond to LGG therapy. So you might wonder how accurate was our ultrasound imaging in measuring these volumes, and this is just a representation of how well we can see on ultrasound imaging. And this is a treatment of an LGG mouse. This is before and after therapy, so you can see extensive tumor infiltration before therapy and after 4 weeks of LGG. Minimal tumor in the bottom and almost no tumor at the top. So then we said, why is this happening? What's going on the sort of mechanistic level with LGG? And we know that previously they looked at CD8 components. So we wanted to look at both cytokine profiles as well as looking at immune infiltration in these tumors and analyze what exactly is going on. So first thing first, we looked at tumor infiltrating lymphocytes in the tissue, which is looking at stroma, as well as the actual tumor itself, and looking at the total lymphocytes count. And really, there was no difference in any of the therapies overall. Then we looked at T-cell infiltration based on stage, and there was a signal in terms of the ratio between the stroma and the tumor, meaning that the higher the ratio, the higher the infiltration within tumor, the lower the stage that these mice were. And then when we looked at gemdosi notably, even though it had a similar tumor TIL ratio, most of the gemdosi actually complete response mice had what we called ulceration of the urothelium, which means there's less of a lymphocytic infiltration and it doesn't act through T-cell dependent response, but rather through an ablative response, which makes sense. So then we looked at urine cytokines using ELISA-based chip technology, which allows you to do up to 15 to 20 different cytokines. The urine was collected from mice after 4 weeks of treatment and then compared across arms. And you can see across all 4 that LGG, even if just grossly looking, had extensive urine cytokine release. And if you just look grossly on all of them, it's really Th1 dependent and inflammatory response that was more overexpressed in LGG compared to BCG. So these are sort of the numbers just to highlight the difference in response. And you can see a lot of the cytokines and chemokines were over-exaggerated in LGG compared to BCG. So then we said, what's actually going to happen in humans when we do this? And obviously we couldn't install LGG intravesically into humans. We did harvest monocytes and T cells using FACS and stimulated them with LGD to see what cytokine profiles these patients were able to produce. And again, what we saw in the presence of monocytes and T cells, these patients were able to have a Th1 dependent response, particularly T cell activation and granzyme release. And then if you just take monocytes without a T cell activation, the response and granzyme release was much lower, meaning there was no T cell activation because there were no T cells present. However, IL-6, which is IL-6 is a cytokine for both innate and adaptive immune system, was over-exaggerated. And then compare that to BCG. Again, presence of T cells and monocytes really ablated any response if you add BCG. So what that means in terms of interpretation is that monocytes alone have a great inflammatory response with BCG. However, presence of T cells, particularly IL-6, is able to elicit a T regulatory response. Sort of, this is the same data. So then we were sort of confused why BCG doesn't elicit much of response as we were expecting in these patients. So what we did is a single cell monocyte type stimulation, which is called single cell secretome, is able to remove any contaminants that you might think in terms of NK cells and any other cells, and you stimulate for 48 hours and you're getting real life cytokine analysis that's based on chip-based technology, which is sort of a novel concept over the last 2 years. So what we found is when we stimulated 1,091 monocytes with BCG, the response again was very minimal, and that was based on GM-CSF and IL-6 response. And again, only 2% of the cells, monocytes, responded to BCG for 48 hours, and most of them only secreted 2 to 3 cytokines, which is a bit lousy. So then we looked at differentiation of cells. If these cells were exposed to multiple cytokines, what actually happens to monocytes? So monocytes have the capability to differentiate into macrophages and also to dendritic cells. Obviously, dendritic cells are what we're interested in, particularly in cancer. So when we compare control, which is monocytes monocytes in media for over a couple of days, compared to LGG stimulation. LGG stimulation did a CD1A, CD11C negative monocytes, CD68 negative monocytes. So that means these are premature dendritic cells. So these are immature dendritic cells that at some point will develop CD11C positivity. If you look at control, most of them, about half, converted to macrophages, and the other half just remained monocytes. If you look at CD8 response compared to BCG, you see that there is a strong CD8 and CD4 response with LGG stimulation, unlike BCG, which is primarily CD4. A couple of things that we have done but are not presented here because of scope of time is that we did a tricolor stain for CD8, CD11C, and CD163. You can see that based on some of the preliminary analysis, the gemdoses has significant dendritic cell stimulation. However, when you look at BCG, there's minimal, and then LGG has significant dendritic and T cell, CD8 T cell stimulation. What we're doing next is we're doing single cell spatial analysis on the tissue, as well as we're currently combining LGD with multiple toll-like receptors to sort of hyper-accentuate the response and potentially get even better clinical response data. To summarize, LGG is an effective treatment for BBN-induced bladder tumor. Our early hypothesis states that it's CD8 T cell response, and it promotes dendritic cell differentiation. It's comparable to intravesical chemo, and it offers a unique cytokine profile both from T and IL-15, which is NK cell marker. I want to thank everyone, particularly Phil, who's a mentor over the last 4 years, and we'll continue to work on together on our microbiome data, as well as multiple members of the lab, as well as multiple cores who are involved in this. I want to thank the BCAN for their support, as well as multiple individuals who have supported me at Fox Chase with additional funding to continue this research, and multiple patients, over 165 patients who have donated their urine for this. And then I look forward to transitioning from a mentee to a mentor to our next generation of surgeon scientists.
That was really fascinating work. Michael O'Donnell, I've done a lot of work with the BCG immune response, and there's always been these missing elements. One of the things we know about BCG is that it both stimulates TLR2 and TLR4 with the TLR4 dominant. There's another mycobacterium that is a killed form of saprophytic, and it's used in India to prevent leprosy. It's called Cadio5. It's a pure TLR2 agonist, and it also potentiates a more of a directed Th1 response without the Th2 elements. So I'm wondering, with these common themes, whether the TLR2 agonistic properties are in fact a key to the success here of the lactobacillus.
You're absolutely right. LGG stimulates toll-like receptor 2. So that's how it essentially works, which is much better than receptor 4 or there's 6 and 5 like other bacteria. So toll-like 2 is the sort of the pathway that it's working from.
In the sense that TLR2 and 4 both converge in a common pathway, how is there a difference? I never understood that.
I think we don't know enough because clearly if we can stimulate 4 and we can stimulate 2 and have different responses, there's probably parts of the pathway that we're not understanding.
Cool. Thank you very much.
You're welcome.
Hi, Jeff Damrauer from UNC. And just taking a step back to the model selection. So you're using a BBN model, which tends to be more kind of a basal subtype. And BCG is used in predominantly non-muscle invasive. They tend to be enriched in FGFR3 mutations, which tend not to be kind of basal tumors if they progress to muscle invasive disease. So could it be that kind of the model selection is dictating why you're not getting a BCG response initially? And may LGG just be a better kind of intravesical therapy because it could be effective against kind of the more basal or luminal non-muscle invasive. So have you tried it in a kind of a more luminal non-muscle invasive setting?
So we've done different cell lines using IncuCyte in terms of the cytotoxicity of the LGG itself in combination with monocytes and T cells, and across the board for multiple JD2s, they're all very consistent responses. However, our next step is to try different models aside from BBN and mice as well.
Hi, Gary Steinberg from Rush University now. You did a lot of microbiome looking at the stuff in the urine. Were you also concomitantly looking at what was in the stool? And do we need to look what's in the fecal microbiome while we're studying the urinary microbiome?
Yeah, we have similar data the 150 patients that collected urine, we also have their stool data. And that data will be presented at the Oncobiome session at the Oncobiome breakthrough session. But it's a little bit different profile for the stool.
Okay, thank you.
Hi, I'm Madhuri from Queens, Canada. Very nice talk and really fascinating work. I was just curious if you used both female and male mice here, and was the data from female mice where we see lack of response?
We used both. We used half and half, so 50-50 because of the differences. And I could tell you more female mice develop what I consider to be a squamous subtype. So the male mice were more likely to respond, but the numbers were, wouldn't call them significant, but they were more likely to respond to this type of therapy. Now, the problem with intravesical therapy in male mice, is that as many of us know who've done installations in mice, is that even though you're putting an angio catheter into the bladder and you're trying to make sure the bladder is filled, I do feel a lot of times the mice get prostatitis because of the sort of the reflux within the prostatic fossa. So I wonder if the mice who didn't respond were just not getting the therapy in their bladder.
A second quick question is, did you co-instill gemcitabine and docetaxel? Or was it separate?
Separate, just like human. Yeah, it was very painstaking. It was very painful. Yes, very painful.
Thank you.
You can thank Max for that.
Laura, excellent talk. Really fascinating stuff, and we're excited to have you come to Case Western. So one of the interesting things that you showed was how BCG didn't really necessarily have the same response, or same mediators of response as the lactobacillus. So would there be any benefit to using lactobacillus to enhance BCG response in terms of like co-installation between the two?
We thought about this as potentially like the next step is, or maybe perhaps not at the same time, but perhaps maybe switching a couple days here and there and do BCG a couple days later LGG, just because we really don't know what happens when the two are combined. So we thought about doing that as well, particularly if we start transitioning to outside the mice. Maybe combination therapy is the way to do things sort of easier down the line. Yeah.
Thank you.
Hi, Neelam from UT Health Science Center, San Antonio. Very interesting talk. I was curious, you mentioned a little bit that the involvement of NK cells in the response that you saw. So I was wondering, have you looked at, like especially with the sorted NK cells from human tissues, to look what kind of effects on the NK cells and what happens to the different subsets? Like, are they activating the dysfunctional subsets, or any hypothesis that you may have?
So when we sorted our monocytes, we did positive selection. So we did 14, 16 classical, non-classical monocytes. The problem is that some of the CD16s will also be NK cells. And then when we saw granzyme with BCG installation out of those monocytes, monocytes don't have granzyme. So we knew that there's a contamination with NK cells, which is how we sort of came up with the idea that the NK cells are actually responding to the BCG. We haven't done anything else in terms of the NK. I think once we do the spatial analysis using single cell data, we'll be able to do clustering and get some of the more additional data in terms of the clusters and the differential expressed genes in those clusters, and get some more NK data on that.
Thank you.
You're welcome. Right. Thank you.
So good morning, everyone. It's my pleasure to introduce the awardees of our Innovation Grant for 2021. So the Innovation Award supports exceptionally novel, high-risk, high-reward projects with great potential to produce breakthroughs in our understanding of bladder cancer. These awards address innovative approaches and new ideas, be they novel theoretical concepts, methodologies, instrumentation, interventions, or other innovations to advance our understanding and management of the disease. So in 2021, BCAN was in the fortunate position to be able to fund 2 awards. So as the past chair of the Innovation Award Scientific Review Group Committee, and I chaired the committee in 2021, I'd like to announce the winners for 2021. So our first awardee is John K. Lee, who is an assistant professor at the Fred Hutchinson Cancer Center in Seattle, and his proposal is entitled Identifying Genetic Determinants and Vulnerabilities of Histologic Variants of Bladder Cancer. Unfortunately, Dr. Lee can't be with us at BCAN this year, and he'll present his work at next year's think tank. Our second awardee is Dr. Weiguo. He's currently a professor of pathology at Northwestern University. So Dr. Kui hails originally from China. He trained as a physician, but soon found his passion in science and biomedical research. In 2002, he came to the US to pursue graduate studies through an exchange program. He undertook postdoctoral training at Yale and then moved to Milwaukee to become an independent investigator at the Versiti Blood Research Institute and the Medical College of Wisconsin. And then just last year, in 2022, he joined the faculty of the pathology department at Northwestern University Feinberg School of Medicine. So Dr. Kui's research interests primarily focus on T-cell biology in the context of viral infections, cancer, and autoimmune diseases. He has made significant contributions to the understanding of how cytokine signals and genetic pathways influence effector and memory T-cell differentiation, as well as T-cell exhaustion. Specifically, he has discovered multiple signaling pathways and transcriptional circuits that regulate the terminal differentiation of effector and exhausted CD8 T cells and the formation of long-lived memory T cells. Overall, his research aims to find new ways to reverse T cell exhaustion and improve control over chronic viral infections and cancer. So I'd like to congratulate Drs. Lee and Kui for their excellent research, and I'd now like to invite Dr. Kui to present a summary of his research, which is entitled BCG-CAR Empowers Adoptive Cell Therapy to Treat Bladder Cancer.
Thank you. Thank you, Dr. Adams, for that very nice introduction. And also thank BCAN to give me the opportunity to present our recent work. As said in the introduction, I'm an immunologist. I have nothing to disclose, but if I throw out jargons, please stop me and we can talk about those right on. You don't have to wait to the end. Let's see if I can learn how to navigate this. So just very nice to follow up Laura's talk and then just give a very quick glance of a view of bladder cancer. And it's the sixth most common, obviously, in the States. And it has a strong sex bias, about 3 times more in men than in women. And then there's a broad range of the type of cancer, and many of you have known this for many, many years. And the advanced age, male, and cigarette smoking are the common factors contributing to the disease. Over the years, as far as the bladder cancer treatment and management has been evolving. And there's obviously the primary treatment is surgical removal, as well as chemotherapies. And in the recent years, immunotherapy came onto the horizon to treat bladder cancer and some other type of solid tumor. Nonetheless, bladder cancer, in my view, probably one of the first and has been longest treated with immunotherapy, which is evidenced by BCG treatment. I'll get into this in a minute. And recently, there's immunocheckpoint blockade and some other type of therapies has come to the new light and has been become the center of many of the basic and clinical research focus, such like adoptive cell transfer therapy. And I'm going to talk to that for the main part of my research presentation. But before I get into that, I just want to give you a little overview what is really happening in the tumor microenvironment and why certain targeted therapies are, basically focus on which part of the approach so that we can achieve the better treatment in the future. So you can take a look at this. Just basically, doesn't have to be the bladder cancer. It could apply to any of the solid tumor. And the tumor microenvironment is quite heterogeneous. But there's 2 major components I want to draw your attention to. Number one is immunosuppressive arm of which consists of myeloid suppressor cells, Tregs, or regulatory T cells. Those things actually suppress immune function. And on the other side, you have functional T cells that supposedly are the tumor-reactive T cells, like CD4s and CD8s. Those T cells are supposed to attack cancer, but they often become dysfunctional. Sometimes they call them exhausted T cells. So the goal of which is here to overcome this, you have to break the immunosuppression in a way of doing this to target this innate immunity part of the equation. At the same time, you want to also recover or restore the function of dysfunctional CD8 T cells and CD4 T cells to allow them to re-energized and vitalized so they can fight tumor back. Then in light of those, like I said earlier, BCG, I have heard about this a lot. Basically, as part of immunotherapy, been practiced more than 40 years in bladder cancer, with this central concept to stimulate the patients or their own immune system, right? And to get them to be revitalized to fight cancer. But this approach mainly targeting the innate arm, means the myeloid cells, and you heard from Laura's talk, they produce a lot of cytokines and then they draw in limited amount of adaptive immunity. Whereas the immunocheckpoint blockade And it says here, and like there's many forms of them, one of which I presented here is a PD-1 and PD-L1 interaction. To block this interaction is vitally important to get the T cells come back to work and overcome their exhausted state so they can fight cancer better. So there are also emerging themes in combined therapies and so forth, but the focus on my talk is actually the following. Oops. There's another type of therapy called adoptive cell transfer. This has been pioneered and spearheaded by Steven Rosenberg here at NIH. The idea is, as I said, there are tumor-reactive T cells. Those T cells are in the tissue and in the cancer site, but they often lose function, so that one can actually, through, for example, surgical resection, take out those T cells, right? And you can in vitro amplify them. That's one of the features of a T cell does, which they can respond to mitogens like interleukin-2 and some other cytokines. They can greatly proliferate and expand in the outside of body or in the bioreactor or culture dishes. And then after you're done with this whole process, there's a polyclonal, meaning they can target multiple tumor-associated antigens or tumor antigens or neoantigens, and you can give them those T cells back to the patients. But you usually require some sort of lymphodepletion, could it be chemo, could it be radio, and then to so-called create space, so the T cells transfer back in or engraft. And those has been shown actually pretty effective in many kinds of cancers, but there are still very predominant, obvious obstacles or limitations to apply this to many types of cancer. Number one is obviously limited number of tumor-reactive T cells, right? You can imagine if you resect a tumor by bladder cancer, you won't get a lot of T cells to work with to start. And secondly, when you put those T cells back, even you get enough, amplify them outside of the body, and then you put them back, they have a hard time to traffic. Their study shows only about 5 to 10% of actually T cells goes back to the cancer itself. And I said earlier also, there's an immunosuppressive microenvironment. You have those myeloid suppressor cells, regulatory T cells, they're producing cytokines, they put out those co-inhibitory receptors. All of those can actually suppress the T cells when they arrive, to limit their anti-tumor function. So with that, we came up with a new idea. So this idea stemmed from many years of our research and thinking. So I used to work in the field of, study T cell memory, effector differentiation, and how this, in response to pathogen infection. So the question here is, many of us here in the audience knows, we don't really die from influenza infection. We can fight off bacterial infection very easily for most part of it. But, the question becomes why we can fight off cancer like we fight off infection. So in mind of that, we basically devised this new idea, or this is part of the, BCAN slogan, like, think outside the box. We should be thinking how to convert or combine this kind of thinking and thinking about something more innovative. So the idea here is because in vitro or genetic editing for T cells become readily available and very easy to do. So what we did here is conceptually, you can think we can get those tumor cells out, right? Again, those tumor cells illustrated here in red, they have antigen specificity, they have tumor reactivity for multiple different tumor antigens. And then we can give them another vector that express another T cell receptor that can recognize antigen from a pathogen or from a vaccine in this case. Now, if we transfer those T cells back, like I showed you before, with this adoptive cell transfer approach, and at the same time, we can apply the pathogen in the same patients or, in our studies, obviously, now this moment, preclinical. And then you can actually amplify those T cells and then draw them to migrate to the site of a tumor, where the trick of this, you have to deliver the pathogen or the vaccine to the site of the tumor. And then also when you're giving those a vaccine, you basically, like BCG, right? I'm going to mention this and some other things. You will very much break the immunotolerance because they have this great ability to elicit the immune response, especially for the innate immune arm. So the prototype of this we have done, to basically test if this thing can work or not. We used actually a very simple, it's almost like a poster child type of cancer in the lab animals, which is melanoma. So this is a B16 melanoma, really difficult to get rid of them. So then here, we take a type of T cells on the left. This type of T cells express a receptor that recognize the melanoma antigen called GP100. And then we program this, give them another TCR, so-called second TCR, to change them to the dual specific. This TCR will recognize the bacteria antigen. It came from a recombinant strain called Listeria ova. So then these T cells will be transferred back to the mice that are bearing this melanoma on the flank, the triangle there. And then now we're giving them adoptive cell transfer with this dual specific T cells. At the same time, we inject a small amount of cell, Listeria ova, into the tumor, intratumorally. Okay. So this is what we found. Turns out it works pretty well. You can see from this slide that no, this is a little hard to present. But on top is the cells only receive the monospecific T cell, means all the T cells only respond to tumor, but not bacteria. On the second row is the mice that receive the dual specific T cells, means that T cells we transferred in can respond both for tumor and bacteria. And then on top, on the column will be like you either treat them with PBS or then inject into the tumor or give them Listeria ova. Well, this become very apparent that only when you're giving dual specific T cells and then at the same time you introduce the Listeria ova to the cancer, you can see great amount of T cell expansion, as shown in that GFP and CY11 double positive quadrant. So then take out those cells, you can examine them, and then you can see they have great amount of ability to kill. They express Grad 9B. They can migrate by expressing chemokine receptors like CXCR3, and then they basically go into the tumor, and then they amount themselves to a large amount, and they can mediate the tumor killing inside of the tumor. This thing stopped working. Oh, there we go. So now this basically just tells you the tumor growth be significantly truncated when you deliver this dual specific T cells, on top of which you give them the Listeria ova injection into the tumor. That's shown in that bottom dashed line in comparison to the all other three groups I showed you. And more, oops, can I go back one slide? Thank you. More importantly, if you see this therapy, we really want to see if we can actually eradicate tumor, right? Like completely get rid of them. So this shows you in the monotherapy group without intratumoral injection in the far left, basically 8 of the 8 miles, they go on for their growth curve, and then you cannot get rid of them, right? That's just basically growth curve. And you look at the other three treatment group, monotherapy with Listeria ova, there's 1 of the 8. They actually accidentally eradicated tumor, which we didn't know what's going on there, but that's just a fact. And whereas you receive dual therapy, but with dual antigen-specific T cells without intratumoral injection of the pathogen, there's nothing happens, as well as just like what you've seen with the monotherapy. Where the striking thing here is the last slide on the far right. Now you're given this dual tumor-reactive T cell versus pathogen-reactive T cell with a dual specificity, and the intratumoral injection with the Listeria ova, 7 out of 10 mice completely eradicate tumor. And we have seen this as well as in the IMB39 bladder cancer, like the similar preclinical data, which we don't show. And this data has been published in 2017 in PNAS. So then at this point, we were really, sort of galvanized and saying, look, can we actually practice this type of things in clinically relevant settings? So this is the concept here because you heard about BCG treatment and the standard therapy in bladder cancer, and it's FDA-approved drug for more than 40 years. And that's probably the best place to start this process. So the concept here is also the bladder cancer often been surgically resected. And then, as I said, you can actually collect those tissue, bladder tissue, and isolate the T cells, tumor-reactive T cells from them, and in vitro amplify them. Now, the only thing we need to do is put those two things together, as I did in those prototype experiments, to devise a vector that will allow the connection or bridging these two things together. So this is where the place where I come to BCAN and then, say, can you fund us for this innovative research? And luckily we got the award. And then just to start with this, to do a proof of principle test, we didn't go all the way, and I'll tell you why, because we want to know if this thing can work or not for first place, right? So we did it very artificially here by implanting a tumor orthotopically in the bladder that will express OVA peptide. And then we can use the same way as I showed you before, just simply give them OT1 T cells, which are a type of CD8 T cell that they can kill, and then give them another TCR that can recognize the OVA peptide. So here we can combine these two approaches together and just prove the principle. You can see the combination of a double cell transfer with the BCG OVA, which is recombinant strain of BCG. And you can see there's great amount of efficacy, and you can see that number of the T cells goes up on the far right again in panel C. So with this, we moved on, and and then you can, again, this is just showing you some of the data that we have that not only they expanded, they went to the tumor, but also they have killer function like measured here by GRAND-MB expression using flow cytometry. So then, technical problem or the barrier, a caveat here is that one can really not practice this with TCR receptor. The reason for that is shown in the left, because the TCR actually consists of the two chains, alpha chain and beta chain, and then recognize the peptide that is presented by MHC, or in humans, HLA, this so-called complex, right? So in other words, my T cell will not work with any of yours and vice versa, because this so-called one key, one lock model. So that created a big problem because you can't really create so many individual vectors to treat every patient. That's going to be too costly. So I turned around because there's a new technology just came to the horizon not too long ago, and you heard about this, has been used to treating heme malignancy like leukemia and lymphoma, called CAR-T therapy. So CAR-T is a chimeric antigen receptor, and this type of receptor did not exist in natural settings. It's a synthetic variant. In other words, it's actually used the part of the feature of antigen recognition of antigen. The B cell receptor recognizing the antigen conformation instead of MHC and peptide complex to as an actual recognition mechanism outside of cells, and then they use the intracellular domain as with the taking from the T cells like CD3, CD28, and co-stimulation of those different generations you can see from the slide. So the key question here is, can we devise a CAR-T vector, and this vector will recognize every time we inject BCG and use it to program the tumor-reactive T cells you isolated from the cancer patients and to treat patients with this type of therapy. So this is how we did it. So to do this actually take a long time, and you need to select the BCL receptor that will have the reactivity to BCG, and then we immunize the mice with this, and we pick a very dominant antigen called AG85B, has been used in developing vaccines, in TB patients and so forth. So long story short, after many rounds of screening, BCR sequencing, and we put the highway chain, light chain, just show you one of the example of this together to create this CAR-T. And then this is one of the screens we have, the kappa chain, which is K, and the highway chain, which is H, and all those things you can see on the slide that we've done multiple rounds of this. And at the end of the day, we pick a couple of those, and then we run on, and the thing stopped working. We started the real experiments. So here is, we're first using the tumor lysis to prime the T cells so that we can ensure this, the tumor specificity for T cells, and then we implant the tumor again orthotopically into the bladder, and at the same time, like wait them to grow. We can monitor them by, I'll show you some of the images in a moment. And then about like a week after the initial implantation, we give them the therapy, which is the adoptive transfer of the T cells, which expresses CAR-T that recognize BCG, AG85B antigen, and then at the same time we get the BCG into the bladder through the catheter. And then now we put those two things together, and then sometimes we give another round of BCG 1 week later, just kind of like what happens like in real human patients. And then we image them like every week, and then to see the tumor progress. I think this is the key part. Oh, there's one more. Sorry, myself did the animation. You can see we can actually monitor the tumor growth, and then on top of which we have mouse patient 5841. This is one of the things that actually you see it is really very gratifying. So by the time on day 0 doesn't mean the state of the implant, but this is like 6 or 8 days after implantation. Day 0 means the time of treatment. And when you give this combined therapy, you can see by day 1, day 8, there's a great amount of reduction. And then after that, like 2 weeks later and on all the time onward, the tumor is completely eradicated. And then if you look at the cohort we have shown there on the bottom right, there are 4 groups of those. You can clearly see their long-term survival, and usually the ones you see like about 50% long-term survival, and those ones, if you look at this imaging similarly as I've shown here, they completely eradicated tumor. So because of tumor the tumor infiltrating immune cells or the tumor itself is so small, so we start to apply for this is a high dimensional method, either single cell sequencing, or in this case, showing you some other means of we have 20 to 30 panel multicolor flow cytometry to characterize the T cell function and the phenotypic changes between different groups. But don't bore you with all the details and all those data we collected, but just quickly summarize what we found. Number 1, we found enhanced T cell infiltration and then great amount of number, as I've shown you before. And then they also have increased effector function, meaning they can kill tumor better, and then they have the tumor site has decreased immunosuppression. That means you have reduced myeloid suppressor cells, reduced T regulatory cells, and collectively that actually account for or contributed to the eradication or the greater control of the cancer. And one thing more important to us is actually given the high recurrence rate in the bladder cancer, we also explored if those animal actually eradicate tumor for a longer time. We isolate their T cells out, and we want to know, do they form memory T cells that really in an ideal world, they will actually reside inside of the bladder and then offer long-term protection for recurrence. So this is still very early on, and you can see from this again, this is probably part of the jargon I mentioned earlier, but they did, take my word for it, express more CD69 and CD103, I didn't show you here, but they downregulate CD6 well. What that really meant was those T cells actually start to reside. They stay in the lumen side, in other words, of the bladder cancer tissue, and then they could offer long-term protection for recurrence, which is hard to do, in experimental settings. We're working out this protocol to test for real if that is the case or not. But in summary, to conclude here, we found BCG CAR vector, which I showed you, can be used as a universal vector that applies to pretty much every situation for reprogrammed polyclonal tumor-reactive T cells. I want to emphasize polyclonal because the tumor continues to evolve. They're going to mutate. So the goal is to target them. Instead of using one target, you should spread that epitope as much as possible. And second point is the BCG empowered REACT. We call them re-energized adoptive cell transfer therapy. Improve the therapeutic efficacy in murine orthotopic bladder cancers. And this new approach increases T cell expansion. I showed you that migration to the site of tumor, their effector function, anti-tumor properties, and then we can also reverse immunosuppression. So by one approach, targeting multiple facets of the immune microenvironment of the tumor. So with that, I'll end by thanking people who did the work. This work is actually started and spearheaded by a very talented former postdoc of mine, Gang Xin. Now he's assistant professor of Ohio State University, running his own lab now. And it's been taken over by another postdoc, Matthew Kudek, and there's a research tag, Wendy Winkler, and Jen Shen, and some other, Ryan Brown, and some other students in the lab. And they've been supported by Varsity Moonshot Project and some other innovation award internally. And our great collaborator, Dr. William C. At MCW, helped us to establish all those models, which is greatly helpful. And we're actually moving forward maybe to talk to some industry partners and to see if we can move forward to the clinical trials. There are some of them actually interested in this and form a startup company to help us facilitate this transition. Last but not the least, I think I want to give a shout out to BCAN, to fund our innovative research. And I pitched this idea to many other places. Obviously, they didn't take it. But BCAN actually, I think, just gave us this great opportunity to move forward with the project. And I'm very grateful for their support. Thank you for your attention.
That's great work. And the idea of giving dual specificity to try to direct more antigen-specific cells, CD8 T cells, so forth, towards the target makes perfect sense. Now, let me tell you, let me just put this in the context of what we already are using. We're already using substances like EV. There have been opportunizumab was looked at because of its expression of certain cell surface molecules common on bladder cancer, EpCAM, Nectin-4, etc.
Yep.
Could you do the same thing here and make a more universal vector without having to add an extra antigen and an injection?
Great question. Yes, that's part of things we're absolutely interested in. Like one of the common mutations, like you're mentioning, is FGR receptor 3. And we're actually now in the process of developing a CAR targeting that specific mutation. And now one can actually using a dual specific or tandem fashion of CAR T and BCG, and you can put them together. So you don't have to go through, isolating T cells from cancer. But I think there's pros and cons on both sides. We're exploring that opportunity. Yes.
Great talk. Congratulations on the results. I'm Roger Lee from Moffitt Cancer Center. So 2 questions. Number 1, do you think that the CAR T cells are actually just engaging the BCG that's in the microenvironment versus those that are actually being taken up by the cancer cells?
Yeah, that's a great question. We actually don't know. But there could be in the in situ. It also could be in the draining lymph node. Either one of those could be true, but we actually don't know.
And then
that's
the
a really
second
good
question
question.
is there any specificity for your receptor to a specific epitope on the BCG, or is it kind of
yes, the one we use by far is targeting specifically for AG85B. It's part of the antigen that expressed on the BCG on the cell wall. and those conserved with actually mycobacteria. So that's why, I mean, this method actually, in theory, can be converted to treat actually TB or, given the conservative region that we're targeting.
And so have you looked at like the surfaceome of the cancer cells once they're being treated with BCG?
Yeah. That's, I mean, no.
Can I
William,
Can
William
I comment
C
on
has
that for a second? So the antigen for BCG, in terms of actually finding BCG and showing tumor cells present BCG, we almost never find it. We almost never find BCG inside the tumor, even though they've been treated multiple times, and we never really find an antigen for BCG.
Okay.
So
that's good to know.
It's
yeah.
probably.
So there's some work from MSK that's shown that some of the BCG particles are taken up by the tumor cells and presented on MHC2 via macropinocytosis. So I wonder if it's a specific epitope from the BCG particle that's being expressed, or do you have kind of some
I think I heard a similar talk. I mean, presentations and publications. Nonetheless, our target in this idea is not really targeting the ones actually pick up the BCG and present it. Rather, the tumor antigen are pre-existing. Those T cells are supposed to targeting those tumor-associated antigens or neoantigens. Yep.
Thank you for your presentation. Armin Smith here. I was just wondering, did you notice effect of the cells on the more invasive components of the tumors, or was it mostly superficial tumors that were coming into contact with BCG?
Yeah, our model is pretty artificial, I would say, although, this is the best we can do to orthotopically establish tumor and then by implant IMB49 cell line, that's like many people do, which we haven't really stratified which stage of the tumor. To my knowledge, many people who do this can correct me if I'm wrong. I think they're pretty much stay at the surface, non-muscle invasive, in other words.
So, yeah, and the reason I was asking is there ability to use this application for like distant kind of tumors, or does it have to come in direct contact?
Yeah, ideally, yes. As I said at the very end, I brush over this very quickly. I think the key is if you have tumor reactivity and those T cells being mobilized, and we know they don't die after the initial, the primary tumor in the bladder has been cleared, they should be able to form memory T cells, and those memory T cells could circulate, could be reside in the tumor. I mean, in the lumen side of the bladder, which can offer greater ability to protect for recurrence or long distant metastasis. That's the concept.
Right. So the idea of establishing memory and long-term protection is very, very intriguing, obviously. And did you test that? If you do a subsequent inoculation, you'll never be able to successful to do that for your cured mouse?
Yes, we're really interested in that. Actually, we're trying to do it. The problem is run into the technical difficulty. It's really hard after many times of doing the catheter and all those in the real other topic model, which is really a technical challenging. I'd like to hear if anyone has better ideas to do this. We have done in the early melanoma model that we've seen the protection for sure. That is established. But that model, again, for bladder cancer is not really a good way. You can't just put you can, but it doesn't really resemble the real situation like what we've seen in humans or in patients.
Yeah, I mean, I think your last point is important because your BCG doesn't work in this model either. And so it's a question here of whether the model is relevant. But I really like the work that you're doing. It's outstanding. I have two questions. One is, do we know that this is, that it's not an off-target effect? In other words, did you use a cell line that doesn't express OVA and show that your T cells are not working good? Yes. And then the second one would be, we've seen really super physiologic effects of OVA-specific T cells. The OVA itself is very antigenic. And so I'm curious as to what you would do for humans. I mean, what antigen, and would this be applicable to a regular antigen other than OVA?
Yeah. So again, the OVA peptide was done only in that pilot experiment where we established proof of principle. Later on, when we did our BCG or dual specific T cell with CAR, and those are the T cells we prime or resemble the TILs that will be isolated from the tumor site, which possess multiple different type of TCRs, and they cover the polyclonal population of T cells, could react to tumor.
Getting back to the discussion, Billy Kim, University of North Carolina. So getting back to the discussion about how this is working and whether it's targeting BCG like in the tumor cell versus not, I think an important question is this BCG specifically driven? And so I know this is a lot of work, but I'm just suggesting this as a control. Yep. If you could just modify the mycobacterium to mutate that specific peptide or a couple of peptides around the antigenic site that your CAR T is recognizing and demonstrate that that therefore abrogates its effect, I think that would really sort of nail down that this is actually dependent upon BCG being present there. So
yep, sure. That's good suggestion.
it's a lot of work. I'm not...
Yeah.
So, a great presentation. So, sort of not to belabor the point about mechanism, but I think we're all very interested in that. So, are you proposing that the mechanism is essentially some sort of antigen spread from cross-reactivity with BCG to the pre-existing tumor neoantigens? And I can certainly understand the merit of this approach and that it would be attractive because you wouldn't necessarily need to sequence and predict the neoantigens. Have you, sort of as the flip side of that is toxicity, have you looked at cross-reactivity of these CAR T cells in your animal models with other tissues and sort of does BCG go somewhere else and then do you get toxicity because of that?
Yeah, that's a great question. Yes, we certainly consider the same lines, and so far we haven't observed any because we're only giving 1 or 2 doses of BCG, and they're 1 week apart. And also we basically took some of the tumor sections and for the histology type of things, we didn't really detect any other things actually spread outside of the tumor or into the other organs. And we haven't done like very stringent measurement like to detect if BCG actually leak into the bloodstream and so forth like that, which we should have done. But that would be some steps we have to do if we want to really move forward for this technology.
Thank you.
Yeah. I missed it. How did you say that the Tregs and the MDSCs were downregulated? What was the mechanism of that?
Oh, they just speak because of BCG injection and some other, this is just an observation. I think they changed the microenvironment of the tumor and then their frequency is reduced compared with the other treatment group.
Okay.
They don't target those cells directly.
Okay, okay.
Yeah, the BCG might, but not the CAR, not the T cell.
In your model, you didn't add anything to try to depress Tregs or MDSCs?
Yeah, BCG itself does that. I think, but the CAR T didn't. Yeah. Thank you. Yeah. You can just yell.
Nice talk. I just want to ask, conceptually, how do you think the CAR T cells are going to survive the hostile environment of the myeloid suppressor cells and the cytokines that are being released by those?
Right.
How do you think?
They don't really, all of them survive. Only when they got to the later tail part, some of them will survive and become long-lived memory cells. And that is not uncommon because similar things happens in any of the infection models. Yeah, there's only about 5 to 10% cells actually can survive and become long-lived memory cells. All right, thank you.
Okay. Thanks.